We are the paradoxical ape. Bipedal, naked, large-brained, long the master of fire, tools, and language, but still trying to understand ourselves. Aware that death is inevitable, yet filled with optimism. We grow up slowly. We hand down knowledge. We empathize and deceive. We shape the future from our shared understanding of the past. CARTA brings together experts from diverse disciplines to exchange insights on who we are and how we got here. An exploration made possible by the generosity of humans like you.
As a background to my talk today, Perspectives on the Future of Fossil-Based Human Origins Research, I want to tell you that I'm a paleoanthropologist who's been conducting field research for the last 30 years in one of the remotest areas of the world and collecting fossils of our ancient ancestors. With this fieldwork experience, I wanted to share with you some perspectives, my hopes and concerns, of course, on the future of fossil-based human origins research. Today, we have thousands of fossils representing more than 20 early human ancestor species. All of the ancestor species older than 2 million years were found in Africa. So when we go back in history, remember what Charles Darwin predicted in 1871 in his book, The Descent of Man, where he said the last shared ancestor of apes and humans lived in tropical Africa. Now, at that time, he didn't have any fossils in hand to investigate, but he was right, and his prediction came to be realized with the discovery of the tongue child in 1924, which is about 43 years after he died. So after the first discovery from South Africa of the tongue child, so many more discoveries were made in so many cave sites within South Africa, and the search for early human ancestor fossils continued further north to Eastern Africa. And a number of significant discoveries were made in the 1960s and 1970s in Tanzania, Kenya, and Ethiopia. So when we look at the distribution of paleoanthropological sites in Africa today, this is what it looks like. As you see on the map, there are sites in Southern Africa, which has been known since the 1920s. And then you have a group of sites in Eastern Africa, which have been known since the 1950s and 1960s. So historically, most of these Eastern African sites have been investigated since the early 1960s. And a number of fossils that are really critical in our understanding of human evolution has been found from these sites. But when we look at how many of those sites were actually discovered since 2000, there are only a few. And the reason why I mentioned that is because that is directly relevant to my talk today. So the fossil record that was available until the 1980s indicated the presence of at least 10 or so species, early human ancestor species, including Lucy species Australopithecus afarensis, which was thought to be the earliest human ancestor at 3 million years ago. And in terms of the phylogenetic relationships, Australopithecus afarensis stood as the ancestor of all the human lineages, one that led to the genus Homo and the other one that led to what we call the robust lineage. And this was as simple as what we could see in the fossil record until the 1980s. So with only 10 species known in the fossil record, our evolutionary history was not really that complicated. But what we see in the 1990s and early 2000s, which, by the way, are usually referred to as golden decades of paleoanthropology, we see a number of new human ancestor species discovered, both from old sites and also new sites, almost doubling the number of known species in the fossil record. Not only that, but we also have the earliest record of human ancestors pushed all the way back to more than 6 million years ago. So this time, our evolutionary history became much more complicated. So in addition to all these new discoveries, we also see advances in analytical methods. 3D imaging became part and parcel of paleoanthropological studies. Various modelings and sophisticated quantitative methods with advances in genetics and also advances in geospatial sciences really helped paleoanthropology to make significant discoveries, new ideas, and test existing hypotheses with all these new analytical methods. But the problem is, all of these advances, even though they've helped us generate more data and provided us with access to more information that we wouldn't have been able to see with our naked eyes, none of these advances helped us increase the actual fossil sample sizes or fill the temporal gaps that are apparent in the fossil record. So this is where I really want to emphasize, particularly in terms of what does finding new sites mean in terms of our understanding of our evolutionary history. Now, the Afar region of Ethiopia has a unique place in paleoanthropology. In this region, we have ancient fossiliferous deposits that pretty much document the last 6 million years of our evolutionary history. And Middle Awash that you see at the bottom here is one example in this regard, where we have a sequence of deposits that sample the last 6 million years. But we will see that there are like some interruptions in the sequence, and without filling those interruptions, those temporal gaps, it would be very difficult to really understand the entire history of our evolution. So paleoanthropologists have been investigating some of the sites since the early 1970s, old sites and also looking for new sites. So we know that Middle Awash, Gona, and Harar have been investigated since the 1970s, and they've produced hundreds of early hominin fossils and also associated vertebrate fossils that tell us about the environment in which these early human ancestors lived. But there are still some gaps in the fossil record that we really want to find by locating new sites, because it's less likely that we will have those gaps filled from the existing sites, especially when we know that paleoanthropologists have been working at the sites for more than 50 years. So finding new sites, like one example that I can mention to you is Ledi Gararu. Ledi Gararu is a site that was discovered after 2000. And now we have the earliest fossil evidence of the genus Homo, which is our genus, from 2.8 million years ago. Previously, we thought that the earliest record of Homo is about 2.4 million years ago. So that's because of the discovery of a new site that we now know that the genus Homo appeared in the fossil record much earlier than previously thought. Another new site that I would like to talk about is Woranzomile, which is where I'm going to spend more time, because that's the site that I've been working at for the last 15 years. So for almost 15 years, I've been participating with the Middle Awash Project as a member, and during these years, I have come to understand the geological history of Middle Awash, the temporal gaps that are missing within the almost 6 million years record of our evolutionary history at Middle Awash. And it's always been my interest to actually see, look for new sites where we can actually fill some of those gaps that we have in beautiful sequences like in the Middle Awash. But of course, I also wanted to have a site where I can work for, as a career in the future. And that's when I decided to do some survey and exploration in the northern part of the Afar Rift. And I spent about 2 years before I finally located Woranzomile. So those two sites are probably two of the most significant new sites that have been added to the older sites within the Afar Rift system. And Oranzomille has a lot of surprises in terms of how we understand our evolutionary history, particularly during the Middle Pliocene between 4 and 3 million years ago. And because of the discovery of this new site, we added more than 200 fossil specimens of early human ancestors, ranging in age from 3.8 all the way to 3.2 million years ago. Although most of these are isolated teeth, we have a cranium that you see on this screen, which is about 3.8 million years old. We have numerous maxillae, the upper jaw and lower jaw, mandibles, postcrania, and a partial skeleton, and including this wonderful partial foot that you see here called the Bertolli foot. Now, each of these discoveries have their own impact on how we understand our evolutionary history. So the work that we have done so far at Oranzomille shows us that there were at least 4 different species identified from the sequences that are sampled by almost 800,000 years. One is Australopithecus anamensis, which has been known from Kenya and Ethiopia, Middle Awash, but it was known only up until 3.9 million years ago. So its discovery at Oranzomille at 3.8 million years ago tells us that anamensis lived longer than previously thought. We have a partial foot from Oranzomille that is really interesting because it shows opposable big toe at 3.4 million years ago, a time when the famous Australopithecus afarensis was roaming, the deserts of, well, not deserts, but the lands in the Afar region. And here we have a species that had kept its opposable big toe and living at the same time in close geographic proximity. Another surprise that we have from Oranzomille is this new species called Australopithecus ramada, which is only known from Oranzomille so far. And then we also have a partial skeleton of Australopithecus afarensis from 3.6 million years ago. Now, Australopithecus afarensis is really well known from Hadar, a site about 30 miles to the south. What is really important about this partial skeleton is that it provided us with a lot more information about some aspects of the biology of and anatomy of Australopithecus afarensis, particularly in the areas of the shoulder girdle and also the rib cage. So this specimen helped us understand better in terms of some of the paleobiology of Australopithecus afarensis. So when we look at the combined evidence, fossil evidence from Oranzomille, what we realize is we're talking about how do we fill those missing stratigraphic gaps that we have in the fossil record. And Oranzomille, one of the things that it did was to fill one of these gaps that existed between 3.6 and 3.9 million years ago. So this was one of the temporal gaps we had in the fossil record, and the discoveries from Oranzomille filled that gap. So this is really important. The second thing is how the partial skeleton of Australopithecus afarensis actually helped us better understand some of the paleobiology of Australopithecus afarensis. And it also happens to be the oldest partial skeleton of an adult individual of Australopithecus afarensis at 3.6 million years ago. From Oranzomille, we also have this new species, Australopithecus ramada, which happens to overlap in time with the well-known Australopithecus afarensis, which is well sampled at Hadar, but we also have a good sequence for the presence of Australopithecus afarensis at Oranzomille. So this new addition is also something that we know because of the discovery of this new site that resulted from survey and exploration in early 2000. But what was really surprising in terms of discoveries from Oranzomille was this partial foot, which again temporally overlaps with what we have identified as Australopithecus ramada and also Australopithecus afarensis. Now, at this time, geological time, about 3.4, 3.5 million years ago, Australopithecus afarensis was the most dominant species. And we wouldn't have expected in a million years that we will find a hominin with opposable big toe at this time. So here is another surprise that finding new sites and finding new fossils brings about. So the Bertolli partial foot is really significant because some hominin species retained opposable big toe as late as 3.4 million years ago. And it also tells us that there were multiple modes of bipedal locomotor adaptations among mid-Pliocene hominins. These are new revelations that we see from Horanzo-Millet. But in addition to increasing the sample size, filling some of the temporal gaps that we have, they also raise some interesting questions in terms of how these early human ancestors coexisted. For example, how did related hominin taxa manage to coexist in close geographic proximity at Horanzo-Millet and not at nearby Hadar? Because at Hadar we have only one species, which is Australopithecus afarensis. But at Horanzo-Millet we have a species represented by the Burtelle foot, another species, Australopithecus deiremida, and we also have Australopithecus afarensis. So we start hypothesizing in terms of what are the ways that these hominids could have coexisted. And we can talk about adaptive radiation. We can talk about how they probably partitioned the habitat and whether they had some differences in foraging strategy and differences in locomotor adaptation and so forth. So this new site opened up not only new questions that we need to address, but also helped us fill some of the gaps that were apparent in the fossil record. And also, another question that comes up from these discoveries is like, which species could be the best candidate for the ancestry to the genus Homo, right? So far, Australopithecus afarensis has always been considered as the ancestor of all species that came after 3 million years. But now that we have multiple species living at the same time with Australopithecus afarensis, we ask this question, then who is the ancestor of the genus Homo? So this is really important discovery. And I mean, after this, I don't think there is any doubt why continued fieldwork research is really important and finding new sites is really important. But you notice that every publication that comes on research on human origins will always end the research paper with more fossils are needed, right? So yes, more fossils are needed because we don't have enough sample for each hominid species that we have in the fossil record. And we also know that there are so many questions that remain to be answered, that remain unanswered, and we need answers for those questions. And the only way we can answer those questions is by increasing the sample size, by sampling larger populations of these early human ancestors. And the only way we can do that is by going out to the field and recovering more fossils. There are also a number of unsampled time periods in the fossil record, and we need to fill those gaps, like what we did with the 3.6 to 3.9 million year gap that was filled by discoveries at Horanzo-Millet. And there is also a need for refining the geological sequences in some of these sites, because we have very good dates for certain horizons, but some of the horizons have a deep time represented between those dated horizons, and we need to refine the stratigraphy accordingly, and that can only be done by going out to the field and doing field research. And of course, field research is really important for students. It gives them the opportunity for hands-on experience, and this is part of, I think this should be part of what they're trained for, like in terms of field work and how it's done. So there are so many advantages for going back to the field, finding more fossils, and also locating new sites that will help us better understand our evolutionary history. So when I consider all the trends that we're seeing today, particularly since 2010, we see that there is this tendency in terms of decline in survey and exploration to locate new paleoanthropological sites. Now, if we don't continue surveying and exploring for new sites, we're not going to be able to document some of these missing gaps in the fossil record. So the second one is we also see decline in funding for field-based human origins research, especially after 2010. There was an increase in the early 2000s or late 1990s, and as a result, you've seen how many specimens, how many fossil hominins were identified in the 1990s and early 2000s. So that was really a good trend, but since after 2010, we see some decline in terms of funding for field-based human origins research. And I also see that training of undergraduate students in the field seems to be declining, and that's not a good sign, like the other ones that I mentioned earlier. So this is also something that we need to think about. So how can we move forward in human origins research. As we know, there is serious problem in terms of like what the trends are, what the current trends are. So the first thing is, I think there should be a consensus among all stakeholders that human origins research cannot happen without the fossils that come from fieldwork. By stakeholders, I'm referring to all paleoanthropologists, students, granting agencies, private and federal granting agencies. All of this are included as stakeholders. So this is something that we have to really understand, that this is a critical part of human origins research. I also think that there is a need to invest in survey and exploration to locate new fossiliferous sites. And of course, we know the logistical challenges and how expensive it is to actually go out and survey and explore to find new localities. But I think the fact that we now have much sophisticated geospatial sciences, we can really incorporate new approaches from those sciences and kind of make the challenges much smaller. And we also need to, as paleoanthropologists, we need to consider fieldwork as a major component of their students' training. Now, as a graduate student, I spent most of my time in the field, and honestly, everything that I know about faunal assemblages, everything that I know about geology, all of it was what I learned in the field, not in a classroom situation. So I believe that having graduate students trained in the field not only helps them understand fieldwork methodology, but also helps them become holistic, all-around paleoanthropologists who understand the context of this early hominids that we are also finding. So without continued fieldwork and recovery of more fossils, without filling the temporal gaps in the fossil record, without refining the ages of the fossils we currently have in hand, I don't think we can fully understand our evolutionary history and how we became who we are today. Thank you.
Thanks for the invitation for this symposium. Today, I'm going to talk about using stem cells to study human origins. So we started this work by asking a question: how is the human brain distinct from other primates? And we know that human brains have increased, neurons have increased dendritic branching. We know that there's increased spine density on the neurons. There is enlarged cortex. And we also know that there is a delayed maturation of neurons in human brains. And that delayed maturation is also called neoteny. So we have observed neoteny in cortical growth, cell cycle length, and dendritic spine maturation, amongst other things. So we wanted to look at systems to understand this aspect of this delayed, protracted neuronal maturation in humans. And there are different systems in which you can look at that, from looking at the DNA to postmortem brain tissues, to fossil evidence, and to archaeological evidence. We wanted to focus on another system in which we use live cells from non-human primates and humans to look at comparative experiments, so we can test and validate some hypotheses regarding neuronal maturation. So stem cells can differentiate into neurons, and we can look at them comparatively between humans and non-human primates and see what are the changes or the differences between cells differentiating. So for this to be a valid system, we need to know if there are differences that are detectable at the cellular level that will be relevant to understanding changes in brain size or neuronal maturation in humans. Are there changes that we can identify in the cell that would be relevant for the individual? And early on, to generate the resources for this work, we used induced pluripotent stem cells. We generated induced pluripotent stem cells from chimpanzees and bonobos, which are our closest living relatives. And we also show that cells from non-human primates, from those apes, were able to differentiate in vitro from the induced pluripotent stem cells, and they expressed neuronal markers that were typical from the developmental stage, such as the ones indicated here, MAP2, TIG1, synapsin, and they also expressed cortical markers. So we were able to generate cortical-like neurons in culture from non-human primates. So a question we had is, and it's one way that we could look at neuronal maturation from this, starting from the stem cells, was to look at neuronal activity. So to detect neuronal activity from the different species comparatively, we used a system called multi-electrode array platform. So that system allows us to detect extracellular voltage changes that are associated with electrical events, here shown by those blinking dots. So those were neurons that are firing action potentials. So our first analysis showed that when we look at earlier during development, humans generate a certain amount of activity, and every trace here corresponds to an activity of a neuron. So that's when a neuron fire, that's why we see a trace. And we compare that to chimpanzees and bonobos. And initially what we see is chimpanzees and bonobos here in red are differentiating more, they were quicker at differentiation compared to human neurons. But later on during differentiation, and that's when you look at 6 weeks now of a neuronal differentiation, those same neurons are now firing more rapidly. So they were differentiating more, they were more differentiated, firing more action potentials and more mature compared to chimpanzees and bonobo. But we noticed that delayed maturation that we also call the protracted development or neoteny in human neurons cultured in a dish. So now we have a system in which we can study neoteny in the dish. So we then ask the question, what are the genetic drivers for neoteny? So can we identify those the genetic drivers that would allow for this delay of human neuronal maturation? And there are different drivers, potentially different genetic drivers. And one of them will be gene duplications. And there are a number of studies over the years that have shown that humans have unique gene duplications that could be responsible for some of the genetic drivers for human neoteny, and other genetic candidates for genetic drivers of human neoteny could be related to gene regulation. So transcription factors bind to specific sites in human genes, and those sites could be evolving different in humans compared to non-human primates. So you still have the same transcription factor, but the binding sites will be different. So we could potentially detect that by looking at differential expression profiles. So there is evidence that there is positive selection of transcription factor binding sites, and that's indicated in this paper. So that suggests that the transcription factor binding sites may be evolving new functions in humans. You can still use the same transcription factor, but the binding sites in the DNA might be different. So with that in mind, we ask the question if changes in gene regulation in humans compared to non-human primates could be a component of human neuronal neoteny. And for that, we looked at differential gene expression in humans compared to non-human primates. And here we used a system that we differentiated the induced pluripotent stem cells that we had from humans, bonobos, chimpanzees, gorillas, and rhesus. And we differentiated them in neuroprogenitors, here called NPCs. And we also differentiated them further into neurons. At different times during differentiation, we looked at 2, 4, 6, and 8 weeks. And then we looked at RNA profiles. So what RNA those cells were producing at those different times during development. So our first question was, are the neuronal progenitors transitioning to neurons in a conserved way? So are we generating human, gorilla, chimp, bonobo, rhesus neurons in the same way. And it turns out that all species do transition similarly from neuroprogenitors to neurons. And we can see evidence for examples of genes here, for instance, CDK1, which is a gene related to progenitors. So when you compare a progenitor, which is a neuronal progenitor that is still dividing, you see that gene that is more upregulated, and it downregulated similarly to all species during differentiation when we're looking at neurons. A similar thing happens to synapsin 1, but on the opposite side. So synapsin 1 is a typical gene expressed in neurons. So we will see it less expressed in neuroprogenitor cells, but when you differentiate into neurons, the expression of synapsin grows. And that's comparable between all species. So we are able to generate neurons in the transitions are very comparable between human and non-human primates. Also, the spatial enrichment of the genes in neuroprogenitors and in neurons is very comparable. So that's telling us that we can make neurons from human and non-human primates in very similar ways. But yet there are some differences. So what are the differences? So one of the differences, and a way in which we can look at those differences, is by doing an analysis called the principal component analysis, using the full transcriptome. And when we do that, so that allows us, this type of analysis allows us to summarize a lot of data from one cell line and reduce it to one dot. So for instance, the triangle here will indicate this, the whole transcriptome of that one cell line. And in that way, we're able to compare a lot of data and summarize it in a figure. So what this figure is showing us is when you compare the full transcriptome from all the species that we tested, the transcriptome recapitulates phylogeny. And that means human is clustering, all the humans are clustering together, the chimpanzees and the bonobos are clustering together, followed by the gorillas, and the rhesus is an outer group. So that's also important to us. That also means that our cellular system is representing to some extent what's happening to the species, to the living species. So what we did then is we performed a weighted correlation network analysis, which is another type of analysis that will identify the top correlated upregulated genes in human-specific networks. So here we're looking for things that are human-specific, for genes that are human-specific and are upregulated in humans. So there are a number of genes that are identified, but one of our top upregulated genes is called GATA3. And we can see here in more detail that over differentiation time, so 0 will be the progenitor, and then we have 2, 4, 6, and 8 weeks, GATA3 goes up during the earlier stages of differentiation in humans, so that's a green trace here, and it stays up over the course of the differentiation compared to the other non-human primates. So we see in this analysis that GATA3 is correlated with genes that are uniquely regulated in humans by looking at the transcriptome. So we got very excited when we saw GATA3 as one of our top regulators, because that manuscript that I showed you before, published a number of years ago now in Nature Genetics, also showed that GATA3 was one of their top transcription factors whose the binding sites were showing the strongest evidence for positive selection. So we wanted to confirm that GATA3 expression was increased. So we confirmed that increase in expression of the GATA3 protein in human cells compared to non-human cells, both chimp, bonobo, gorilla, and rhesus didn't express GATA3. And we also showed that in postmortem brain using Allen Brain Atlas, we showed that GATA3, we pulled the data showing that humans have increased expression of GATA3 compared to rhesus. So both on neurons that we were culturing the dish, as well as in brains, postmortem brains, GATA3 was all upregulated in human cells. So we noticed that GATA3 is increased in human in vitro and in prenatal brain. Now we wanted to know, so since GATA3 is we are proposing that it's a human-specific regulator of certain sequences in the genome. One way in which we can look at what GATA3, which genes GATA3 is binding, is to use a technique called chromatin immunoprecipitation, followed by sequencing. So that technique allows us to look at which genes, areas, which sequences in the genome, the transcription factor of choice, which in this case is GATA3, which areas GATA3 is actually binding to in the genome. So when we do that in our cell lines, we notice that there was an enrichment of GATA3 on promoter regions and 5' UTR of genes, which is something that would be expected as a characteristic of a transcription factor. And we notice a number of genes, and here is an example of a gene called Notch2NL that has enrichment of GATA3 signal at the 5' UTR. Additionally, we noticed that there was a spatial enrichment of GATA3 bound genes on the area of called subventricular zone when you looked at 15 post-conceptional week-old postmortem human brain, meaning that GATA3 bound genes are also enriched in that area where neurons are, newborn neurons are differentiating into, migrating and differentiating into more mature neurons. Additionally, we noticed that the binding sites we detected using that technique called chromatin immunoprecipitation, followed by sequencing, those sites were located in areas in the neurons, and they are under positive selection in humans. We use that same algorithm that Arbiza et al. In Nature Genetic used in the past. So with that experiment, we concluded that GATA3 binds to unique sites in human neurons that are under positive selection. We wanted to know what knockdown of GATA3 would do, and we wanted to ask the question if it would be inhibiting human-specific expression. So we generated cell lines that were using SH RNA against GATA3. We decreased the expression, we're able to decrease the expression of GATA3, and we wanted to, both in protein and RNA, we wanted to look at what would be the consequences of that. So, here is that same type of plot, principal component analysis, where we are specifically, we included all species, but we are only looking at genes that are identified as GATA3 regulated within humans. So when we look at a principal component analysis, including only this, the genes that I mentioned that are GATA3 regulated, we can see that humans are clearly separated from the other non-human primate species. Now, what happens when we now plot human cells in which we have decreased expression of GATA3? So look at what happens now. The black symbols change their position in principal component analysis, especially in principal component analysis 2, so the y-axis, to a location that is more similar to where bonobos and chimps, non-human primates, are located. And another way to visualize that is to plot the same values that are plotted on the PCA. Those are called eigenvalues, and we and show them separately what is happening on PC1, which is the y-axis, and PC1, which is the x-axis, and PC2, which is the y-axis. So while PC1, there is not much change. So the black box plot are the cells that have been treated with SH against GATA3. So decreasing GATA3 doesn't change the profile much. When you look in PC2, what you see is there is a clear decrease of the box plot that is related to SH that was treated with SH GATA3, and now it's comparable with chimpanzees and bonobos compared to where it was before GATA3 treatment. And that we also wanted to show some examples of human-specific expression patterns that are expressed more similar in non-human primate samples after treatment with SH GATA3. So SH GATA3 is able to change expression of some of the genes, the lack of GATA3 changed the expression of some of genes to a more similar expression to what you see in non-human primates. And here's an example of TMEM260 and CT and NAL1, where treatment with SH GATA3 changes the expression of the genes to more similar to the levels that are expressed in non-human primates. And we have also other examples of genes that shift in human expression to a more pan-like state after knockdown. So with those experiments, we are suggesting that GATA3 regulates a component of the transcription that distinguishes human neurons from pan neurons. Our other question was would that, now that we notice that there is a change, do we see any differences in the physiological maturity rate in human neurons? And that we have to remind us, what we have seen before is that a protracted development, maturation in human neurons. So human neurons here in green are delayed maturation compared to chimpanzees and bonobos. We have shown that before. And what we we're looking here is the same parameters, which is the mean firing rate, as well as bursts, which are indicators of neuronal maturity. And when we look here at the red squares that I'm highlighting, you can see human neurons treated with SH against GATA3 compared to untreated or humans treated with SH control. You see that there is an increase in activity in those neurons, and we can see a plot here showing in black an increased activity for both parameters of the mean firing rate as well as number of neuronal bursts, both parameters that indicate neuronal maturity. So the absence, removing GATA3, speeds up the maturation of human neurons. So GATA3 presence acts to delay the rate of maturity of spontaneous action potentials. And we think that this is a key aspect of human neoteny. So GATA3 is involved in that process. So to summarize and conclude, I hope you saw evidence that GATA3 is uniquely upregulated in human neurons in comparison to other non-human primates. I hope you also understood that GATA3 is bound to genes that display human-specific dynamics. GATA3 binding sites are under positive selective pressure in human neurons. GATA3 regulates a component of transcription that distinguishes human neurons from pan, and GATA3 acts to delay the maturity of spontaneous action potentials, and that's a key aspect of human neoteny. And to conclude, we think that our findings underscore the importance of identifying how human-specific gene regulation is brought into an already functional transcriptional network of an organism, potentially through the coevolution of transcription factor binding sites. So the idea that if you can still keep the same regulations, but if you change the binding sites or you have new binding sites, you're able to change important aspects of neuronal maturation. And with that, I would like to acknowledge our collaborators, Rusty Gage from the Salk Institute, Caterina Semendeferi from UCSD, and people that were more directly involved with this work. Sarah Linker is the lead author. Renata Santos are also very involved. And we also had support from Inigo Narvaisa, Mayan Wang, Amandeep Sharma, Anna Mendes, Ruth Offner, and Lynn Moore. And thank you so much.